So, ladies and gentlemen, you are all welcome to our today class, and of course, for our today's class, we are going to continue with enzymes. We are going to continue with the concept of enzymes activity and enzymes specificity. So let me share my slides. So I hope you can see my slide. Can you all see my slides? Okay. So thank you very much. Actually, you know that we have started with the concept of enzymology, and one very interesting things that you should understand that I sent you, I think, three video clips on the aspect of enzymology. And please, if you know that you didn't watch those videos, please go back and visit them. It is very, very important. And as you know that you are going to start writing your exam very soon, and of course, this concept of enzymology is also the content cover a lot. So that is why we have to try as much as possible to see what we can do. And as I promised you, I wanted to at least, maybe if possible, maybe by tomorrow, let's have another class so that to see how we are going to cover with the aspect of the enzymology. And in addition, I think yesterday some of you I sent one very short video on the aspect of transport of molecules across the cell membrane, and I will continue like that, especially for this next maybe for the at least like to 6 days. I will try as much as possible to summarize all the content that we do, all the content that we treated, maybe in just 5 minutes, to have them in 5 minutes, so that at least you will have a summarized content to help you easily remember what you have learned during our classes. But I advise you, for those of you that did not actually join our membership to the YouTube channel, please try to do that, because that is where I'm going to post all those videos. And in addition, I will also try to get like five different years to see how I'm going to help you and solve the question paper, and I will upload it to the channel for the members to watch it. So if you know that you are not part of the membership, please try to as much as possible to join the membership. And I think the concept is clear now. Or is there anyone that have any question, any complaint, or any problem? If there is, let me know before we actually start the class. Is there any issue, any challenge? Okay, how can I join the membership? Okay, what you are going to do, just go to the channel, you will see where you are going to click subscription. So I know that you already subscribed. So you will see where you will see join. So click on that join and you proceed. Then you can easily become a member. So if you know that you are not a member, try to join as a member. And I know that for the issue of your lectures, I know that I have done my best and I'm actually almost done with the lecture. So if you are not part of the members, we can try to join. So try to do that. Okay, so As I said, enzymology. So you see, as we started, that enzymology is the study of enzymes. And what are enzymes? Enzymes are biological molecules that speed the rate of the chemical reaction. So whenever we are talking about enzymes, is when you have the substrates and then the enzymes will convert it to product by lowering the activation energy. Generally, most of the other, generally, even if there is no enzymes, there are a number of reactions that they can still convert the substrate to product, but it may actually take time. It may be years. So that is why sometimes it is not advisable to understand or to carry out any kind of reaction, especially in biochemistry, without using enzymes because it will delay. And even in our body system, if there is no enzymes, a number of reaction will not take place. So that is why it is very, very important to know the aspect of enzymology. And also, as part of the class that we are going to do, I will try as much as possible to see how I'm going to introduce you to the aspect of clinical enzymology. The aspect of clinical enzymology. So, in today's class, actually, we are going to actually focus more on the aspect of enzyme specificity. We are going to focus more on enzyme specificity. So, what is enzyme specificity? When we said enzyme specificity, it's just trying to tell us how enzymes is actually having the ability to identify a one single substrate, enzyme substrate. So, and enzymes always try to be very selective. Enzymes is always trying to be very selective. So now what I want you to understand is that when we said enzyme specificity, it means how enzymes will be able to identify a single substrate. So how is that possible? So in enzyme specificity, each different type of enzymes will surely act only on one substrate. So one enzyme cannot act on two different substrates. It is highly specific. So each and every enzymes or each and every type of enzymes is able to identify one specific substrate because each of these specific enzymes or each of the enzymes are specific because they have different shaped active sites. And of course, when we said enzymes active sites, it is a region where a substrate bind and convert it into product. Okay, Favor, check very well. If you actually subscribe, check very well. You will see where you are going to join. So, yes, that is what you are going to do, Amina. So, what we are saying actually, each and every enzymes have a specific active sites, and these active sites have different shape. So the shape of the active site is what determines which of the substrate can bind to, because each enzyme is specific to a particular substrate. So enzymes bind with the substrate in the active site. So because different enzymes have different active site, that is why different enzymes act on different substrates. Like for example, now look at the image here. Look at in the enzymes one here. Look at the active site of the enzymes. Look at the active site of the enzymes. You can clearly see that in the active site of the enzymes, it is somehow circular. It is somehow circular, and this is where the substrate bind to. So you see, the shape of the active site is complementary to the shape of the substrate. If you look at it very well, it's complementary to the shape of the substrate. And that is why, like these enzymes, A can only recognize and bind to these substrates. And look at the enzymes too. In the enzymes too, if you look at the active site, it looks like triangular in shape. So it means that this substrate with the triangular shape cannot fit into enzymes 1. And that is why enzymes 1 cannot act on the substrate 2 here, because the active site of enzymes 1 here is not complementary with the substrate 2 here. But the substrate 1 here is complementary with the active site of the enzymes 1 here. And that is why it can catalyze the activity, or it can convert the substrate 1 here to the product based on the complementary side of the enzyme. And look at also uh, enzymes 3 and the substrate 3. You see, the substrate 3 is hexagonal in shape. And of course, it is also the active side of the enzyme is also complementary to the hexagonal shape of the substrate. And that is what you call enzyme specificity. So enzymes is able to only recognize and catalyze one substrate. And why? Is because each and every enzymes have its own specific active sites. Okay, ladies and gentlemen, I have this question. Remember last time I asked you this question. So I don't know if someone can answer it. In fact, in some of the videos I send you on enzymes, I also talk about that. Active site is made up of two sites in it. In the enzymes active site, it is made up of two different sites. Please, who can mention that? I'm waiting for the answer. Who can mention the two other sites that are found in the active site of the enzymes? I'm waiting for the answer. You know, no answer? So in the active side of the enzymes, there is what we call catalytic site and the binding site. A binding site is also part of the active site where a substrate binds to, while the catalytic site is a site on the active side of the enzymes where the substrates are catalyzed into product, and that is what we call catalytic site. So enzyme is made up of binding sites. Sorry, the active side of enzyme is made up of binding sites and the catalytic sites. So ladies and gentlemen, this is the most important thing that you should understand under enzymes activity. So enzymes activity, sorry, enzyme specificity. Enzymes specificity is able to catalyze only one substrate based on the different active site that they have. So then the next thing, what are the different types of enzyme specificity? There are four different types of enzyme specificity. Number one, we have absolute specificity, we have group specificity, we have linkage specificity, we also have stereochemical specificity. So stereochemical specificity is made up of optical specificity and geometric specificity. So these are the four types of enzymes specificity. So we have absolute specificity, group specificity, linkage specificity, stereochemical specificity. So under stereochemical specificity, we have optical specificity and geometric specificity. And then, so now let's look at the different functions of each of these enzyme specificity. So we are starting with the absolute specificity. So we said absolute specificity, it means that enzymes can recognize only one of substrate and implement their catalytic functions. Like for example, uricase only hydrolyzes urea to form ammonia and carbon dioxide, but not methyl urea. So it means that in the absolute specificity, means that enzymes can only be able to recognize one type of substrate and catalyze that substrate. Like for example, urea and methylurea, they are almost the same thing. It's just that the difference in methylurea there is methyl group attached to it, while in the urea there is no methyl group. So enzymes ureas can only hydrolyze urea but not methylurea. So that is to show you how specific the enzymes is. So that is in the case of absolute specificity. And then the next thing after absolute specificity, as we said here, the enzymes will recognize only one reaction. And then, so the next one is actually group specificity. So what is a group specificity? A group specificity, of course, is when an enzymes can only act on a molecules that have a specific functional groups, such as amino acid, phosphates, or methyl groups. So it is only active, it only recognizes, or it only acts and is only specific to a particular functional groups. It can be amino acid functional group, it can be phosphate functional group, it can also be methyl group. So all this is actually under group specificity. So it's only specific and recognized as well as catalyze a specific functional groups, and that is why it's called specific. Sorry, that is why it's called group specificity. And then the next thing is we also have what you call linkage specificity. So what is a linkage specificity? Okay, Bala Salamatu, you are welcome for joining as a member to the channel. So the next thing after linkage specificity is that, sorry, the next thing is linkage specificity. So what is a linkage specificity? The linkage specificity is when an enzymes act on a particular type of chemical bond regardless of the race of a molecular structure. Assuming now you have an amino acid, so in amino acids, remember in amino acid there is a peptide bond. So peptide bond are usually formed between amino group of one amino acid and the carboxylic of another amino acid. So if you have an enzymes that recognize and catalyze only the carboxylic and amino group of the peptide bond, irrespective of the other groups that are present in the chain. That is what you call linkage specificity. Linkage, where you have a bond. So the enzymes will only act on the chemical bond, irrespective of what is there in the other part of. So, ladies and gentlemen, you should understand this. So, one absolute specificity is when only one enzymes catalyze only one substrate, one reaction. Group specificity when enzymes is only specific to a particular, or when the enzyme is only specific to particular functional groups, and that is what we call group specificity. And there is also a linkage specificity. So when you say linkage specificity, in the case of linkage specificity, the enzymes only act on a specific chemical bond, irrespective of whatever other molecules that are present in the compound. And then the next one is stereo. Someone is saying that they are not seeing the slide. Okay, so let me share it again. Let me share the slide again. So I hope you can now see the slide. So in the case of stereochemical specificity, is actually when enzymes will only be able to recognize a particular isomer. It can be, the isomer can be stereoisomer, it can be optical isomer, or it can be stereoisomer, it can be geometric isomer, it can be optical isomer. So when you have an enzymes that is only be able to recognize a specific types of isomer. So that types of specificity is called stereochemical specificity. So it's only applied to a specific isomer, and that is what you call stereochemical specificity. Okay, Grace, you are welcome to the membership. So this is what you call enzyme specificity. So enzymes that are very specific on the substrate that they catalyze, on the substrate that they combat. One enzyme is only specific to one substrate, and one substrate is only specific to another. So this is actually what we call enzyme specificity. So and the enzymes is specific to a particular substrate based on the active site. So and of course from our previous slide we discussed that the active site is what actually determine which of the substrate will bind to. So different enzymes have different active site, and that is why they are able to recognize different types of substrate, and that is what you call enzyme specificity. And the enzyme specificity are divided into four: absolute specificity, group specificity, linkage specificity, and stereochemical specificity. So in the case of stereochemical specificity, the first thing that will come into your mind is optical isomer and geometric isomer. That is, the enzymes are able to only the enzymes. Sorry, so the enzymes will only be able to recognize a specific isomer. It can be stereo isomer, it can be optical isomer, it can be enantiomers, it can be geometric isomer. So in stereo specificity, we are talking about the specificity of an enzymes. And there is also a linkage specificity. So in linkage specificity, we are talking about a chemical bond, when an enzymes will only be able to recognize a specific type of chemical bond. It can be peptide bond, it can be glycosidic bond, it can be phosphodiester bond. So that is in the case of linkage specificity. And in the case of group specificity, it's about a functional groups. And in the case of absolute specificity, is actually about how one enzyme will only be able to recognize only one substrate. Okay, Okere, you are also welcome to joining. You are also welcome for joining our membership. So this is what you call enzyme specificity, ladies and gentlemen. And I hope it is now clear that to understand that enzymes is only specific. So that is why, because enzymes they are generally specific, if someone is developing a particular disease, like for example, in my last class, I talked about an enzyme called angiotensin converting enzyme receptor 2. So angiotensin converting enzyme receptor 2 is an enzyme that convert angiotensin 1 to angiotensin 2, and angiotensin 2, remember, is actually a vasoconstrictor. So if you are able to, in most cases where you have people with cardiovascular disease, or people with so many other types of diseases, so the best way actually to control that disease, if it is actually as a result of if the pathogenesis or pathology of the disease is due to enzymes, is to get a compound that will inhibit that particular enzymes. So that is what you need to understand. So like for example, in the case of angiotensin enzymes or uh, angiotensin converting enzymes, it converts angiotensin 1 to angiotensin 2, and angiotensin 2 is a vasoconstrictor. So of course, if you can get a particular compound that can block the enzymes, it means that angiotensin 1 will not be converted into angiotensin 2. It means that you lower the risk of developing hypertension to that particular individual. So once you block that enzyme, there is no any other enzymes in that particular organism, or in that particular patient, or in that particular human, that can come back again and now convert angiotensin 1 to angiotensin 2, because it's only angiotensin converting enzymes that play that role. And if that enzyme is actually blocked, so the substrate cannot actually be converted into product, and that is what happened in enzyme specificity. So if enzyme is meant to catalyze this substrate to product, and the enzymes is now blocked, so it means that the activity or that enzyme sorry, that substrate cannot be converted into product except in the case of isozyme. Except in the case of isozyme. Isoenzymes or isoenzymes. When you said isoenzymes, are enzymes that are one, they may have different amino acid sequence because enzymes are protein and they may be present in different location, different part of the cells, but they convert the same substrate to product, and that is what you call isoenzymes. Isozymes, that is, are enzymes that are be able to catalyze the same types of reaction, but they may actually come from different locations. They may come from different angles, and that is what you call enzymes specificity. Enzyme specificity, if this one is converting this to this, so that is what you need to understand under enzyme specificity. Ladies and gentlemen, I hope it's clear. Are you understand, are you okay with the class? I hope you get it. Rukayat Al Amin, you are welcome for joining the membership. Is there any question before we proceed? Okay, if there is a question you can ask your question before you proceed. Is there any question? You shouldn't forget that absolute specificity, the enzymes act only. Okay, sir, I have tried unsubscribing for to see the membership and I didn't see it. No, check very well, favor. You don't need to unsubscribe. You will have to subscribe and you will see join. So, Funsho is asking about the isoenzymes again. When we said isoenzymes, although we are going to discuss that too, but when we said isozymes, they are enzymes that catalyze the same substrates into product. They may be more than two different isozymes and they may have different locations. Like, for example, lactate dehydrogenase is an enzyme that convert pyruvates to lactates. And that enzymes, we have some of the enzymes, we have one of the isozyme in the liver. We have another one in the muscles. We have another one in the heart. And all of them, they are converting substrates, sorry, they are converting pyruvate to lactate. But they are of different location, but they catalyze the same types of reaction. And that is what you call isoenzymes. That is what you call isoenzymes. They catalyze the same type of reaction. They catalyze the same type of reaction, but they are of different location. That is what you call isoenzymes. That is what you call isoenzymes. So I hope we show you are now okay. So, and then the next thing that we are going to talk about is enzymes activity. So what is enzymes activity? Enzymes activity is defined as the amount of enzymes that is required to convert 1 micromole of substrate to product per unit time under specified conditions. So what does that mean? Ladies and gentlemen, you should understand that when we are talking about enzymes, we have enzymes convert substrates to product. Enzymes convert substrates to product. So when we said enzymes activity, it's trying to talk about amount, how much of the enzymes is required to convert 1 micromole of substrate to product per unit time. That is what you call enzymes activity. How much of the enzymes do you need, or how much of the enzymes is required to convert substrate to product per unit time? That is what you call enzymes activity. So enzymes activity is defined as the amount of enzymes that are required to convert 1 micromole of substrate to product per unit time under specified assay conditions. That is what they call an enzymes assay. The word enzymes assay usually is a qualitative test and it can also be quantitative. It can be qualitative and it can be quantitative, but it depends on the types of the assay. What do we mean with the enzymes assay? When we said enzymes assay, is just when you are trying to check whether you have a particular enzymes in a given solution or not. So to know whether you have an enzymes in a particular solution or not, or whether you have an enzyme in a particular sample or not, that is what we call an enzymes assay. To check the presence or absence of a particular enzymes in a given sample is what we call an enzymes assay. But in the case of enzymes activity, it's just to know the amount of the substrate that you are needed. Sorry, the amount of the enzymes that you are needed to convert 1 micromole of the substrate to product per unit time under specified condition. And that is what you call enzymes activity. And this enzymes activity have a formula, and the formula for calculating enzyme activity, it can be the amount of the, because remember, whenever you are actually whenever reaction is taking place, whenever there is a reaction that is taking place, you should understand that in the reaction usually the substrate uh, is converted into product. So now what happens if you are converting substrate to product? Ezema, you are welcome to the membership. So, in the reaction, usually we have what you call decipherence, because if you are producing A to B, you are converting A to B in case of chemical reaction. So, what is happening? As A is converting to produce B, so the concentration, that is the amount of A, will be decreasing, while at the same time the amount of B will be decreasing because it's A that you are converting to B. So A is disappearing and B is appearing. And that is what we call actually enzymes activity. So to measure the enzymes activity, you can measure the enzymes activity based on the amount of the substrate that disappeared per unit time or amount of the substrate that appear per unit time. And that is how we calculate, that is how we calculate enzymes activity. The amount of the enzymes, sorry, the amount of the substrates that are disappeared per unit time or amount of the products that appeared per unit time. And that is what we call enzymes activity. So you should know that if you want to measure the activity of the enzymes, you are always monitoring either the disappearance of the substrate or appearance of the product. Like for example, now how do we do some assay? You may have, maybe you know from the literature, you know, or maybe if you know that there is a general knowledge that enzymes 1 usually convert a red substrate into blue product. A red substrate into blue product. So how do you know that you have an enzyme? Assuming now you are given a solution, assuming that you are given a solution that you were told, you are, or maybe you were asked to check whether you have an enzymes, whether you have an enzymes in the solution, and you know that that enzyme generally convert red substrate. Let's say the substrate, let's say it's an organic compound and it is red in color. You have an organic compound and it is red in color. And you know that maybe from the literature, the red colored product, sorry, the red colored substrates usually it is converted into blue color. So how do you know that in the solution you have that enzymes? So you do, or you actually do what you call enzymes activity assay. So how do you do the enzymes activity assay? You get the blue, so you get the colored the red colored substrate and you add it inside the solution that was actually suspected to contain your enzymes of interest. You incubate it and you allow it to see if the red color is deciphering to produce the blue color. So what will happen if the enzymes is there? It means that as the reaction starts, as the reaction starts, the red color will be disappearing. It will be disappearing, while at the same time, the blue color will be appearing. So when the reaction reach completion, all the red color will disappear, and then the blue color will be completely appear. So that is how to do enzymes activity, or that is how to test for the activity of an enzymes. Is that clear? So how do you calculate the activity? Then you calculate how much of the red color disappear per unit time, or how much of the blue color form per unit time, and that is how to calculate enzymes activity. And in some of my next videos, I'm going to actually show you how to calculate enzymes activity. Okay. So it can be, you can measure enzyme activity based on the color change, based on maybe bubbles formation, maybe based on formation of precipitate. So many other things you can do that to actually check enzymes activity. So you see, 1 unit. When we said 1 unit, it means 1 micromole per minute. 1 unit, that is one enzyme unit, stand for 1 micromole per unit, and is actually the SI unit of an enzyme's activity. So if you want to measure the enzyme's activity, it's usually measured in micromole per minute, and one international unit is equivalent to 1 micromole per minute. And this unit is the international unit of enzymes, and the SI unit of enzymes activity is the ketal. So you can use also ketal as an enzymes, sorry, as an enzymes activity unit. You can use ketal. And what is ketal? Ketal is defined as the amount of enzymes that are required, or that is required to convert 1 mole of a substrate to product per unit time, and that is what we call enzymes ketal units. So enzymes activity has a unit as ketal, or 1 micromole per minute. All these are the enzymes units. So for the ketal, it's defined as the amount of the enzymes that is required to convert 1 mole of substrate to product per unit second. Per unit second. So now the next thing that we are going to look at is a specific activity. So what is a specific activity? A specific activity of an enzyme is defined as a number of enzymes units per milligram of protein. So it's also measured in micromole per minute per milligram. But what is the difference between enzymes activity and specific activity? There is also a formula for calculating specific activity. To calculate specific activity is going to be enzymes activity divided by the protein concentration. So enzymes activity divided by the protein concentration will give you the specific activity of an enzymes. Is that clear? Okay, Habila, you are welcome to the membership. So class, I hope you are all clear with the concept. Is that clear? So that is the specific. Yes, sir. I'm trying to join and have but actually you didn't join. If it joined, I would see it here. So this is what we call enzymes activity. So the specific activity of an enzymes. So you can also measure the specific activity of an enzymes by actually measuring the activity, or the enzymes activity divided by the the total protein concentration, and that would give you specific activity of an enzyme. So the next thing that we are going to look at is enzymes turnover number. What is an enzyme turnover number, or KCAT? KCAT stand for the enzyme turnover number, which is defined as a minimum number of molecules of substrates that are converted to products per active site per unit time when the enzymes is saturated with substrate. So in the case of enzyme turnover number, it means that it's a maximum. It's a maximum number of molecules of substrate that are converted to product per unit active site per unit time when an enzyme is saturated with the substrate. And the enzyme turnover number, which is KCAT, are usually calculated using Vmax. When you said Vmax, it means maximum velocity divided by E of T. And of course, this E stands for enzymes concentration, and Vmax stand for maximum reaction rates, and then the KCAT stand for turnover number. So the turnover number is equal to the unit of enzymes activity divided by the mole of enzymes. So in the case of turnover number, is the space is actually a specific activity or is enzymes activity divided by the number of mole of enzymes, and that is the formula for calculating turnover number. And then the next thing after turnover number is maximum velocity. So maximum velocity is the velocity obtained under a conditions of substrate saturation and under the saturation of the enzymes and specified condition of pH, temperature, ionic strength, and Vmax is constant for a given enzymes. So when we said maximum velocity, you know that actually when we are talking about enzymes, enzymes convert substrate to product. So how fast the enzymes is converting the substrate into product is what you call the rate of the reaction. So they call the rate of the reaction. So generally, if you have an enzyme, so you are increasing the concentration of the enzyme. So generally, what happens that this amount generally what happens if you have an enzyme and you have a substrate, so the active site of the enzymes is usually bind with the substrate. So as you are increasing the concentration of the substrate in the active site, then the activity of the enzymes will be increased. So there will be an increase in the enzymes activity. But in a situation where the enzymes is completely filled with the substrate, meaning that there is no more substrate to be accommodated. Sorry, there is no more space in the enzymes active sites to accommodate more of the substrate, then the speed or the rate of the conversion of the substrate to product will remain constant. So if it reaches that stage, so that stage is called maximum velocity. It means that there is no more increase in the rate of the reaction. And that is what you call maximum velocity. So ladies and gentlemen, if you have any question concerning what we discussed now, ask question. And if there is no any question, then we are going to share another PowerPoint on enzymes kinetics. So ask question if you have any question, you can ask. Is there any question? Do you have question? So if there is no any question, we are going to, I'm going to send another link, then we all join again. Thank you.